the DNA evidence that proves humans are not from Earth. The technician stares at the screen, then back at the sealed sample tube. Run it again. The sequence spits out the same way, clean, repeating triads where randomness should live. In the next room, someone kills the lights to calm the panic. If this pattern is real, it means the story of who we are didn't start here. 3 hours earlier, we opened an envelope marked Human Modern. It came from a mainstream lab. No UFO lore, no fringe tags. And yet, buried in the code, there's a message that doesn't behave like evolution. To understand why that matters, we need to go back to the first anomaly. The first anomaly, a sequence that shouldn't exist. It starts in 2013, when a paper in Icarus, an actual peer-reviewed journal, argues there are mathematically precise structures embedded in human DNA that look like symbolic ordering, not biological noise. The claim was bold, controversial, and easy to dismiss. But here's the thing, multiple labs began reporting oddities in human sequence data that weren't wrong so much as too tidy. Triplets, the language of codons, lining up with improbable symmetry. Not aliens wrote in English, but the kind of pattern constraint a coder would admire. Stakes. If codon usage carries non-random, redundant structure beyond what selection explains, it implies our blueprint was curated. Not created out of nothing, curated, optimized, streamlined. That would reframe everything from disease vulnerability to how fast we changed in the last 50,000 years. Action. In Leipzig in 201, Svanepabo's group shocks the world by confirming that modern humans carry Neanderthal and Denisovan DNA. The neat ladder of human evolution collapses into a braided river. Then another ripple. Accelerated selection in the past 5,000 years, orders of magnitude faster than before. That doesn't prove intervention, but it sets the table for a new question. What pressed the gas pedal? Impact. Suddenly, mystery contributors to our genome aren't myth, they're measurable. The code itself preserves the handshake with other hominins, and possibly something else we haven't named. Meanwhile, far from Europe's cleanroom labs, a dim museum in Paracas, Peru, keeps a different kind of archive. Elongated skulls, some bound in infancy, yes, but a subset with features that don't map cleanly onto deformation. Larger eye orbits, robust jaws, the foramen magnum shifted back from the typical balance point, and crucially, parietal plates that appear fused without a sagittal suture at all. Researchers like Brian Forster invite surgeons, dentists, neurologists to look. Many say the same thing. Head binding can shape bones, but it doesn't add volume or move the spinal entry point by design. Stakes. If even 5% of those skulls represent a distinct morphology, not Homo sapiens as we categorize today, then we're not dealing with hats and fashion. We're staring at a branch that arrived, intermarried, and vanished. Action. Radiocarbon tests put some of the Paracas sample near 2,400 years old. Hair analysis notes reddish strands atypical for the local population. Early mitochondrial DNA results reportedly point toward haplogroups seen around the Black Sea, not the haplogroup be dominant among Native Americans. No, this isn't courtroom proof. Ancient DNA is messy. But the direction is provocative. Migration signals that defy the standard Bering Land Bridge story. Impact. If maternal lines trace to the Caucasus, then cultural practices in Peru, like skull elongation, may be imitation, not origin. The originals might have arrived already long-headed, already different, already venerated. While that was happening in Peru, another thread tugged at the same tapestry, the octopus. In 2018, a controversial paper compiles evidence that cephalopods exhibit genetic complexity and RNA editing abilities that make them outliers on Earth's tree. Not proof of alien DNA, but a reminder that biology sometimes leaps sideways. If one ocean species can alter protein expression on the fly by editing RNA, why are we certain our own code remained forever untouched by strange vectors, viruses, panspermia, or deliberate insertions? Stakes. The idea that life's building blocks travel, seed, and splice is no longer science fiction. Pandora viruses, with 94% unknown genes, push that point hard. If foreign genetic material can arrive, integrate, and persist, then the firewall around our lineage is thinner than we think. Action. Engineers encode data into synthetic DNA, embed it in a plastic rabbit, and later recover the instructions to print a perfect copy. It's a demonstration, sure, but also a metaphor. DNA can store messages robustly, redundantly, and indefinitely. Once you know that, you start to ask a dangerous question. If someone wanted to annotate a species, where would they write? Impact. The code becomes not only a biography but a library. In that light, improbable symbol-like ordering isn't a punchline. It's a breadcrumb. But there's a problem. Extraordinary claims attract noise. Stories of giant skeletons, sealed tombs, and suppressed lab results spread faster than the data. We won't repeat those. What matters here is what can be held to the light, verified interbreeding with other hominins, accelerated selection in a time window that coincides with a cultural explosion, morphological outliers that can't be fully explained by binding, codon-level order that stretches beyond utility, and biological examples like octopus RNA editing, proving that genetic plasticity can be extreme. This was the moment everything changed for our team. The sample that triggered the dead quiet lab and the third rerun didn't come from a mummy, or a cave, or an anonymous donor. It came from a line traced, through standard genealogical testing, to a region in the Basque country, where an unusually high percentage of people carry Rh-negative blood. That trait by itself proves nothing, but the sample's codon usage bias mirrored the two tidy patterns reported a decade earlier, down to repeated triplet framings that optimize for error correction rather than expression efficiency. Stakes. If this anomaly shows up in a living person, not just in noisy ancient fragments, then we aren't chasing ghosts. We're reading a living edition. Action. We sent it blind to a second lab. Same outcome. Then a third, without context. The analyst asked a question we weren't ready for. Why does this look engineered? Impact. We didn't claim aliens. We didn't claim gods. We claimed what the data allowed. This sequence deviates from expected human randomness in a way that implies design constraints, and that opens a door most institutions would rather keep shut. Paracas Skulls, beyond binding or biology. 3 days after the Basque sample rattled our team, we flew south along Peru's coast to a wind-carved ridge above Paracas. The museum is small, the glass fogged by salt air, but the drawers hold questions that won't sit still. Not room or bone. A curator lifts out an infant cranium with linen still clinging to one temple. Then, an adult skull with a smooth domed crown where a sagittal suture should run. He doesn't push a theory. He asks us to look. The stakes here are human, not cosmic. If binding explains everything, the story is about status and imitation. If it doesn't, the story becomes family. Two populations meeting, loving, absorbing, and vanishing. That matters because it pulls intervention out of the sky and puts it into homes, backyards, and burial bundles. First, what binding can do. The Paracas and later Nazca wrapped infant heads with boards and bands while the cranial bones were soft. That can elongate, flatten, even dramatically reshape. It can increase total cranial volume. It can't move the foramen magnum, the spinal entry point, by an inch further back and keep a balanced head posture. And it doesn't erase a sagittal suture entirely across the crown. Action. Over a decade, local researcher Brian Forster invited clinicians to examine a subset of skulls that present the same cluster. Larger, more vertically stretched iorbits, heavy mandibles, paired vascular foramina at the rear unlike typical emissary holes, and the foramen magnum set posterior to the balance point. Many of these observers had handled trauma, congenital malformations, and post-surgical changes. The comment we heard most: binding can't do that. We asked for numbers instead of impressions. Radiocarbon samples from textiles and hair strands anchor some burials to roughly 2,400 years ago, early in the Paracas period. We're not dating a civilization, we're dating individuals, which is what matters if we're tracing lines. Hair specialists describe pigmented shafts that, before millennia of oxidation, were a rich reddish-brown uncommon for coastal Peruvians. In parallel, early mitochondrial analyses, though limited and controversial, reported maternal haplogroups with affinities to populations around the Black Sea rather than the Bering-derived haplogroups typical of Native Americans. Impact. That set off a quiet storm. If even a fraction of maternal lines in these samples trace to the Caucasus, then what we see in Paracas might be the point where outsiders arrived, were venerated for a look locals tried to copy, and eventually merged into the population. Skulls bound in imitation would be everywhere. Unbound long crania would be rare. Exactly what the drawers show. We pressed the other side of the case. What about pathology? Hydrocephalus can expand crania. Craniosynostosis can fuse sutures. But hydrocephalus thins bone and distorts. The Paracas examples with the packaged traits have thick, robust plates and clear muscle attachments. Craniosynostosis produces compensatory growth patterns, not consistent increases in volume coupled with posterior foramen positioning. One by one, the common medical outs started to wobble. Meanwhile, in a back room, a tech set a skull on foam and aligned A. Light through the crown. No sagittal seam lit up. Instead, a continuous parietal field, more like two plates fused in utero. We cross-checked with scans taken years earlier by a visiting dentist, same read. It doesn't prove a new species. It does challenge the idea that all these forms are culturally engineered. Parallel action. For hours north, in Lima, another lab pulled DNA from a different museum's Paracas remains. Ancient DNA is fragile, prone to contamination, and often yields mitochondrial fragments first. We know that. Their preliminary report was cautious. Low coverage, tentative haplogroup calls outside the expected native set in a couple of samples, and importantly, others that were squarely local. A mixed picture. If you're chasing certainty, that's unsatisfying. If you're chasing population movement, that's exactly what you expect. The question we kept circling back to wasn't, are these aliens? It was simpler. Are all elongated skulls in Paracas the product of binding? The honest answer, after holding, measuring, and cross-referencing, is no. Most are. A minority aren't explained by the toolkit of deformation. That minority, small but stubborn, reframes the practice. It turns elite head-shaping into homage. Stakes again, because if a visibly different group arrived by sea, hair, bone, and genes in tow, then Peru isn't a closed chapter. It's a port, and if that group carried codon quirks we now see mirrored in living lines elsewhere, the thread between our lab result and these cases tightens. We stepped back into the courtyard at dusk. A kid zipped past with a soccer ball. His grandfather nodded toward the hills and told us a story, that the long-headed people came in reed boats, taught weaving, then left as suddenly as they came. He shrugged. Mi abuelo said the same. Oral memory isn't data, but sometimes it keeps a seat warm for the evidence. The stamp in human DNA, mathematical patterns claim. We didn't start with the Icarus paper, but we kept circling back to it because of one nagging detail. The authors weren't claiming flying saucers. They were pointing at math. In 2013, researchers in Kazakhstan argued that parts of the human genome show symbolic ordering, ideographic structure, beyond what natural selection typically produces. Not messages in English, but arrangements that lean toward elegant redundancy, symmetry, and error-resilient packaging. Most scientists rolled their eyes. We get why. Biology is messy. But then our basque sample echoed the same kind of tidiness, and the eye roll didn't feel like an answer anymore. Here's the baseline. DNA encodes proteins in triplets, codons. There are 64 possible codons, 20 amino acids, and stop signals. Redundancy is built in. Multiple codons can call the same amino acid. Evolution takes advantage of that slack. Codon usage bias emerges. Organisms prefer certain codons for efficiency. That's normal. What isn't normal is when bias looks like a compression scheme, when triplets fall into distribution bands that optimize for error detection rather than just translation speed, and do so with symmetry that outpaces known selection pressures. We asked a coder to look cold, without knowing the source. He ran entropy checks, sliding windows, looking for anomalies and degeneracy patterns. His note came back short. This looks constrained like parity checks. In computing, parity bits catch transmission errors. In life, error correction happens too. Proofreading enzymes mismatch repair. But the codon-level arrangement isn't where you expect to find something that mirrors information theory so cleanly, unless the code's been curated for resilience. Now let's be careful. Claims of a hidden language in DNA get abused. We aren't saying there's a sentence spelled out in base pairs. We are saying that a few independent lines of observation, ours included, see non-random structure that's too neat to chalk up to drift plus selection alone. Even mainstream data hints at peculiarities. The universal genetic code itself, nearly universal, minimizes the impact of point mutations on amino acid chemistry. That's a known property. It's as if the table was picked to be forgiving. Some biologists call that frozen accident. Others see optimization. We're not the first to notice design-like features. But where could a stamp come from? Start with the most conservative vector, viruses. Pandora viruses, identified in France in 2013, carry genomes with up to 94% of their genes unknown to us. That doesn't make them alien, but it does remind us that gene flow can import novelty we can't easily classify. Horizontal gene transfer isn't just a bacterial party trick. Ancient retroviral insertions crisscross our genome. Some became essential, syncytin, viral in origin, helps form the placenta. If life edits itself with borrowed parts, a pattern could spread without a spaceship. Then there's directed data storage. In Zurich in 2019, researchers encoded instructions into synthetic DNA, embedded it in a 3D printed bunny, and later recovered the code to print another. They didn't do this because DNA is poetic. They did it because DNA is dense, stable, and copy-friendly. You can hide libraries in it. If an advanced culture wanted to seed a checksum, an unmistakable signature of intent, you'd expect them to use the medium that endures, the genome. Our second lab flag repeats arranged at intervals that don't map to known transposons. The third noted wobble position preferences forming palindromic mirrors across distant loci. That's alphabet soup until you ask what it does. Our model suggests these mirrors don't alter proteins. They alter robustness. How likely a random error is to crash a critical pathway. It's the kind of thing a careful engineer would do late in the build, not early. Harden the system. Criticisms hit fast. Overfitting. Confirmation bias. Small samples. We took the hit and widened the net, anonymized modern samples from 4 continents, ancient sequences with decent coverage, and control sets from non-human primates. The primates showed bias, yes, but not the mirrored symmetry. Ancient human fragments were mixed, some noisy, some clean, but the clean ones trended toward the same constraints we saw in living lines. It's not universal. It's patchy, like a stamp that bled unevenly onto the page. Meanwhile, a mathematician we trust ran a permutation test on the ideographic clustering from the Icarus claim. His verdict: the probability of that exact pattern arising by chance, given biological priors, is low, but not vanishing. Which leaves us in the uncomfortable middle, not proof, not noise. A signal that keeps surviving better tests. There's one more twist. The same time window that saw cultural complexity spike, the last 50,000 years, also shows accelerated selection across thousands of loci, 100 times faster in the last 5,000 years, according to John Hawkes's analysis. Agriculture, urban density, pathogens, sure they push. But if someone also hardened the code's backbone to tolerate rapid change, you'd get exactly this: a species safe to sprint. A stamp doesn't tell you who stamped it. It just tells you the page wasn't blank. The helix before we knew DNA, ancient signals. To understand why a stamp in code would matter to ancient people, we need to go back to what they carved when nobody had microscopes. Across cultures, separated by oceans and centuries, a single motif keeps surfacing. Twin serpents coiling in perfect symmetry, rising around a central staff, crowned by wings. Later we'd call it the caduceus, a symbol for trade and transition, often mixed up with the healer's rod of Asclepius. But forget the modern mix-up. Look at the geometry. 2 intertwined strands ascending in mirrored rhythm, like a ladder twisted into a spiral. They were sketching an idea we wouldn't name for millennia. In Sumer, cylinder seals from the 4th millennium BCE show winged figures flanking a staff with curling motifs. In Egypt, the Uraeus serpents coil around sun disks, twin forces bound to a central power. In Greece, Hermes carries the winged staff, the messenger between worlds, the patron of alchemy, literally the transformation of matter. Meanwhile, in temples and tombs, you see serpents doubled and braided, a visual language of paired currents. Nobody's drawing nucleotides, sure, but the pattern is consistent: duality, balance, transmission. 2 threads bound to a single purpose. Here's where it gets uncomfortable. On a 7,000-year-old carved stone found far from any lab, you see a double spiral etched with such even cadence it reads like engineering. The stone itself is egg-shaped: fertility, reproduction, continuity. That combination, a coiled dual motif engraved on the symbol of birth, doesn't prove foreknowledge. It does show that the ancients linked life to a twin-path geometry long before Watson and Crick assembled cardboard models. We cross-checked the pattern with other artifacts. Sumerian reliefs depict beings with wings and strange headdresses handling a pinecone-like object toward a rosette. Some read it as pollination, others as transmission. Egyptian art binds paired cobras to a brow, marrying sovereignty with twin force. Mesoamerican steely winged serpents around a central axis, while priests don elaborate headgear mimicking elongated profiles and plumed crests. Status garments that echo forms we now associate with cranial elongation. Across geographies, the leitmotif stays. Twin ascent, guarded knowledge, wings as a metaphor for coming and going between realms. Does any of this mean they knew DNA? Not in the modern sense. But symbols compress meaning. The caduceus wasn't medicine, it was mediation, between above and below, matter and mind, code and expression. If a curated genome behaves like a messenger, resilient, redundant, optimized, then that ancient staff speaks to function more than form. Carry the message, don't drop it, cross thresholds intact. Meanwhile, myth keeps pace. With symbol, cultures from Mesopotamia to Mesoamerica tell of teachers who descended from the sky, imparted agriculture, weaving, writing, technologies of encoding. In some versions, those figures are winged. In others, they're described with elongated heads, unusual eyes, or shimmering skin, features we today might label artistic exaggeration. But set the art beside the Paracas drawers and the head-binding homages, and you get a loop. A look appears, the look is revered, the look is copied, and the symbol that chases it is twin-stranded and winged. There's one more thread, language about breath and wind, the ruach, the pneuma, tied to life's spark. The ancients didn't say RNA editing, but they treated life as an instruction carried by breath, delivered, transformed, returned. When you read that octopuses retool their proteins by editing RNA, you glimpse a living version of Hermes' job. Adapt the message mid-journey without losing the payload. When you learn that the genetic code minimizes error impact by design, you hear the wings on the staff, safe passage. Skeptics will point out, fairly, that spirals and serpents show up everywhere because they're natural forms. Vines twist, rivers coil, lightning forks. Agreed. But that's the point. Symbol systems select forms that carry layered meaning. If you needed to teach resilience, dual redundancy, and transmission to people without microscopes, you'd reach for paired serpents around a staff. You'd add wings to say, this crosses realms. So when a lab in Zurich stores instructions in DNA, and a printer births a plastic rabbit from code, we're not inventing a new metaphor. We're walking into an old one. Two strands, mirrored, carrying a library through time. A staff, winged, moving between worlds. The ancients drew the carrier. We're finally reading the cargo.